Welcome once again. My name is Liev Sosa, and I am a trained medical doctor who now works as a quantum health coach, and I am enrolled with Quantum University in order to accomplish and finish my certification. So it is a pleasure for me to share this with you. So why 3-video series? So why should I learn about genomics? Why should I care about genomics? So the main goal is that you understand the basic concepts of epigenetics, and you will learn very soon what that means, which is exactly the same that your genes can be turned on or off according to your environment. And we're going to learn also what is the environment. So you want to do that in order to understand about your body and take control of your biology, which is what we have as main goal as bio-optimizers and biohackers. So the second video is about neurotransmitters and why should we know about neurotransmitters. So it's because our genes, they determine how we produce and how our neurotransmitters work, and our neurotransmitters, how we make them and how we break them down is what actually shapes our personalities. If we are more on the accelerated versus on the slow pace, it is determined by neurotransmitters. And neurotransmitters are molecules that are in a complex network that is constantly moving, and it can be precisely understood, and your genetic data will help us craft a lifestyle plan that is in agreement with how you are made naturally. So we work with your genetic makeup as opposed to trying to be something else that may not be what you are naturally meant to be. The third video is the endocannabinoid system, which is the largest endorphin network in our bodies. So endorphins, for those who don't know, is anything or body chemical that is related to feel good, to relieve anxiety, and to put us to sleep, and to relieve our pain. So the same substances do all of those functions. So the endocannabinoid system deserves attention because it is a very healthy way of supporting our overall well-being and homeostasis, which is our balance inside our body that will always be in interaction with our external environment. So, moving on, we're going to start with the first video, which is genomics. And we want you to learn about it because integrating and applying your genomics data will allow you to know what you need in order to choose in order to maintain a balanced brain into longevity. So, the table of contents. So you're going to understand what genetic material is: DNA, a gene, and a chromosome. You're going to also learn the difference of the approach of genomics versus genetics. We're going to introduce you to the concept of a SNP, or single nucleotide polymorphism, and we're going to talk about environmental considerations and conclusions. So what is DNA? DNA, or deoxyribonucleic acid, is a self-replicating material present in nearly all living organisms as the main constituent of chromosomes. It is the carrier of genetic information. So we all have DNA, and as you can see here is the very famous double helix that has 2 pairs of nucleotides and 4 types. There's 4 types of nucleotides for the DNA, and there is 4 types of nucleotides for RNA. So we are going to take a look at those two and see one side comes from mom and one side comes from dad. Okay. So where is the DNA located in the cell? So definitely in the nucleus of the cell. That's where we have most of our chromosomes are organized there and protected. But let's not forget that we also have DNA in the mitochondria. And this DNA is relevant to the cellular behavior, how the cellular, mechanisms for the, inflammation, detoxification, etc. So all of those functions are also regulated by DNA that is in the mitochondria. And what is important to understand is that DNA in the mitochondria is not organized and protected the way it is in the nucleus, so it is more vulnerable to radiation and negative toxins that actually can affect our DNA. What are genes? So a gene, as you can see, is a segment of DNA of this double helix sequence that builds, has the information to build one protein. So that's exactly what it is. It's a segment of the chromosome of DNA or RNA which encodes for a molecule that has a function. Most of the times it's proteins, but it's also more complex than that. Now, this double helix information, it encodes, or it actually organized and folded in itself into structures called chromosomes. And the chromosomes have arms, short arms and long arms, and the centromere in the center, and they also have telomeres at the end of each arm. And we have 23 pairs of chromosomes, and as we said before, they are mostly organized in the nucleus. So what is genetic information? The information encoded in the DNA sequence that will allow for the synthesis of a protein. So this is exactly the same, but here what we show you is that the replication of the DNA leads to a transcription process into RNA, and from the RNA we go through the translation, which actually leads to the synthesis or the making of that protein. From the point of view of the approach of traditional medicine and health, when we talk about genes, it's often static. And when it is unfavorable, we tend to say that, it's due to poor genes and there's not much that you can do about it. But over the past 20 years, we have learned different. So let's go through the difference between genomics versus genetics. And then we're going to continue down the rabbit hole of the genomics. So I hope that so far, so good, and that the information is clear. If you have any questions, you can enter them in the chat box and I will be very happy to answer them. So what are genetics? Genetics is the science that studies heredity, role and function of a single gene and its impact on acquired traits or diseases. Single gene towards acquired traits or diseases. Now, what is genomics? So genomics has its roots in genetics, and it is the science studying the structure and function of all genes in the human genome and the interaction between genes and environmental factors. So this is totally different. As you can see, it's actually inclusive and integrative of all the genes of the human genome and how they interact with the environment. And that's very interesting. So all of this research started back in the 2000s. The Human Genome Project started in the 90s, and it was finished in 2003. Actually, it was started. I can't remember exactly when it was started, but I know that it was finished on 2003. And what we learned from the Human Genome Project is that we were actually expecting to have a lot more genes than creatures that are more simple than us. And what happened is that we figured out that we, with 23 chromosomes, 64 chromosome, 23 pairs, we have millions of genes, but basically we don't have more genes than a worm. We have thousands of genes, not millions, thousands of genes. But what we understood is that, we didn't understand, we had the idea that there was the exactly same amount of DNA to determine what the animal will become, but we had a lot of DNA that they say that didn't do anything, and they call that junk DNA. And there was a lot more questions that were asked, and this actually gave birth to new research, like the 1000 Genomes Project Consortium started right after the end of the Human Genome Project, and it was finished in 2015. So this actually went through a lot of data, and they did a lot of genome-wide association studies. And they could see that, what a SNP does is that it provides diversity for our species, and it's the reason why our skin color is different, our hair texture is different, our personalities are different, and we get sick with different problems. So it's all about, it's the same genes, but the function is what is affected. So this really helped us understand the similarities and the difference of a person's susceptibility to chronic diseases. And they did that because they actually went through different experiments that proved that, what really determines the behavior of the cell is not what DNA, if you actually remove the DNA or put it back, it doesn't really make that much of a difference. It was all determined by the conditions of the environment. So basically, like in the mid-2000s, the epigenetics and all this genomic revolution really started since the early 2000s. And we understood that genes can be turned on or off according to environmental factors. So there's psychogenomics, there's pharmacogenomics, there's oncogenomics for cancer. So there's been use of genomic sequencing for all kinds of wellness purposes, and also to predict prognosis and to change the course of certain diseases. So there's a lot of research going on. So what is a SNP, or a single nucleotide polymorphism? So now that we have this background information, it's really easier to understand what it is. And you can see here on this image that we have this sequence of cytosine 1-in that actually it's this, like the one on the top. The only thing it takes for us to make a SNP is actually to change just one of those pairs. So it's a single nucleotide. The nucleotide is 2. And when you change just one, it's still the same gene that makes the same protein, that makes the same function, but not exactly the same. So it can alter the gene expression and the function of the protein encoded by the gene. So if we say that a chromosome which is how, there is all this information organized, we can make the analogy with a computer library. And if we take a segment of that DNA from the chromosome, we actually have genes. So out of these two genes, this gene here encodes through this sequence for this protein, collagen. So we can say that the genes, so the DNA are the files and the genes are documents, is what makes sense of the files. All right? So now, if we change this sequence or this nucleotide, what's going to happen? So what happens is that instead of getting the word collagen, we get the word collagen, which sounds almost the same, but it's not quite. So how does that manifest from the function point of view? Is that you can have, for example, if you have a collagen O1 snip, that means that we have increased risk for osteoporosis, that we have delayed wound healing, that we may have accelerated aging patterns due to poor skin firmness, because these are what the functions of the collagen proteins are in our body. So if we have a SNP, so it's still collagen, but it's a collagen that works less well. So that the same applies into neurotransmitters and mental health. And that's why we say that if we know where the weakness is, then we have better, we have the plan to actually intervene with precision. That's exactly what we mean. So we are talking about turning on or off different genes according to the environment. So what is the environment? The environment is everything that is not you. Your environment is the food that you eat. It is the cosmetics that you put into your body. It is the water you drink every single day. It is the people around us. It is the Wi-Fi, the computer, the cell phone, our bus, the workload. All of those things are the environment. And when we receive those triggers or stimulations from the environment, so that is when we actually turn off or on certain genes. So let's say that I am working for a company and I work 12 hours per day on the phone. I have no time to take care of myself. Then after working long hours, I like to stay up because I have to live a little bit. So I'm living on an overworked kind of lifestyle, an overworked lifestyle that is favoring the expression of most of my stress hormones. So when we perceive danger, and we perceive danger if we are not sleeping well, if we are, in an actual dramatic situation that could compromise our physical integrity, for example. So any of those issues are going to raise our stress hormones, which is going to make that we are tired, that we gain weight, that we are not really having any desire for sex or anything related to that. So that is genetic expression in the context of stress. Now, let's say that November arrived and I was totally exhausted from work, but I have holidays. So let's imagine that we could still travel and I take a flight 3 hours down to Cuba and I go see my family and I have good food, and above all, I meet the sunlight. And the sunlight is going to make me make vitamin D. And if I make vitamin D, then I can make my serotonin and be happy. Then I can have my dopamine and feel stimulated and motivated to go for hikes and go do different things. So my skin will be clear, my everything will actually change because we just changed the environment, going from a working stressful environment onto a sunshine holiday destination. Our genes are expressing all the time. Our immune system is all about genetic expression. When we, all of our functions in our bodies, we have millions of chemical reactions that happen in our body at all times, like all the time we're alive, right? So all of that is subject to be turned on and off, and that is what we call the modifying or modulating of the expression genes. And we know that there are certain genes that are very important that regulate the expression of others. And one of the most important is actually vitamin D. So that's why, being a person who is dark-skinned, imported into Canada, because I was not born here, I really, really, really appreciate the effect of the sunlight when it's there. And since I use light therapy during the dark, long winter months, I really feel that the winter effects are really, really less. So that's what I want you to understand as manifestations of genetic expression, as something that is dynamic. It's always happening. And that's why we say that health is a journey. So we are arriving to the conclusions, and we can say that your SNPs are like genetic glitches that need fine-tuning in your brain and also in the rest of your body. When your SNPs, which are weaknesses, right, or genetic glitches, are exposed to specific environmental triggers, is that trip to Mexico, it is that ankle fracture, it is that moving into a new home with all different kinds of paint, smells. So all of those environmental triggers is what challenges our genetic expression. Okay? And then our neurotransmitters imbalances appear as a reaction to that when we are having actually low reserves to start with. Then when the root cause of those imbalances is not addressed, then diseases arise. And I must say that we all have SNPs that can potentially manifest as risk or benefit, and by default, we are all awesome. And with the right support, we can thrive, most of us, and we can have a mix of different interventions that can actually make it that you feel good. So there is hope, and we want to share that with you. There is always hope. Early awareness is key to preserve your vitality instead of waiting and reacting to later manifestations of disease. And then the last conclusion is that your genes can be turned on or off according to your ability to manage your lifestyle, which is how you conduct yourself in your environment and how you adjust to it. So that's all. I hope that this has been helpful for you and that it will help you understand how powerful it is to know exactly what neurotransmitter you don't produce optimally and how just with a tiny bit of support at your own pace and rhythm, you can be amazing, because we all are. Thank you very much, and I will see you on the next video.